The absence of blood vessels, neuron fibers, and rod cells in the fovea lead it to be the area of most acute vision. Cones are responsible for color vision. Each cone contains one of three types of iodopsin pigment, and each type of iodopsin is sensitive to one of the three primary colors of light, being red, green, or blue. The model of trichromatic color vision proposes that all colors perceived by the eyes are a combination of the three basic colors to which the cones are sensitive. The brain then interprets this combination of different types of cones that have been stimulated as the different colors we see. Because cones are less sensitive to light than rods are, they require larger quantities of light to stimulate them. As a result, cones function best in high intensity or bright light, giving daytime vision. Cones take longer to regenerate once they have been bleached by light. The effect of bleaching of the rods and cones is experienced when one is exposed to a blinding flash of light. For example, when somebody takes a photo and the flash goes off, we cannot seem to see anything for a brief period, which is usually only a fraction of a second afterwards. And once these photoreceptors regenerate, then we're able to have normal vision again. So the next dot point says identify that there are three types of cones, each containing a separate pigment sensitive to either blue, red, or green light. So this is just an identified dot point. We don't need to go into huge amounts of detail at this point. We just need to know that there's three different types. Because all rods have only one type of pigment known as rhodopsin, they are not sensitive to different colors. Rhodopsin is a broad spectrum pigment. Its peak sensitivity is in the 500 nanometer wavelength region of the visible spectrum, but rods do not allow us to perceive any color. Vision involving rhodopsin in rods only allows us to see shades of black, white, and gray. We'll be looking at a little bit closer at these a little bit later. Each cone contains one of three types of iodopsin pigment and is therefore most sensitive to light in one of three wavelengths. These pigments result in cone cells being sensitive to the short wavelengths of blue light, which have a peak sensitivity at approximately 455 nanometers, or medium wavelengths of green light with a peak sensitivity being at approximately 530 nanometers, or the long wavelengths of red light with a peak sensitivity being approximately 625. The sensitivity of a particular cone cell allows it to detect light to some extent on either side of these peak sensitivity, giving an overlap of the colors detected, as we can see here. Red cones are actually more sensitive to yellow light, as we can see in this section of the graph, than they are to red, but they respond to red light before any of the others do, therefore behaving as red receptors. So light of any particular wavelength may stimulate more than one cone. By comparing the rate at which the various receptors respond, as well as the overlapping colors detected, the brain is able to interpret these signals as intermediate colors. This allows us to see a variety of colors. As we can see in the table, the cones stimulated by different colors of light the different cones stimulated by light create different colors that we are able to detect. So if the red cones are stimulated on their own, we see red. If only the green cones are stimulated, we see green. If only the blue cones are stimulated, we see blue. However, if the red and green cones are stimulated of equal value, we see an orangey yellow color. If red and blue are stimulated, we see magenta. Green and blue, we see a color called cyan, which is sort of like a teal. And if red, green, and blue, if all our cones are stimulated, then we see white. This is just a short video that explains how color is detected by our brain. So we'll just have a watch and then we'll move on.
You might have heard that light is a kind of wave, and that the color of an object is related to the frequency of light waves it reflects. High-frequency light waves look violet, low-frequency light waves look red, and in-between frequencies look yellow, green, orange, and so on. You might call this idea physical color, because it says that color is a physical property of light itself. It's not dependent on human perception. And while this isn't wrong, it isn't quite the whole story either. For instance, you might have seen this picture before. As you can see, the region where the red and green lights overlap is yellow. When you think about it, this is pretty weird. Because light is a wave, two different frequencies shouldn't interact with each other at all. They should just coexist, like singers singing in harmony. So, in this yellow-looking region, two different kinds of light waves are present. One with a red frequency and one with a green frequency. There's no yellow light present at all. So, how come this region, where the red and green lights mix, looks yellow to us? To understand this, you have to understand a little bit about biology, in particular, about how humans see color. Light perception happens in a paper-thin layer of cells, called the retina, that covers the back of your eyeball. In the retina, there are two different types of light-detecting cells, rods and cones. The rods are used for seeing in low light conditions, and there's only one kind of those. The cones, however, are a different story. There are three kinds of cone cells that roughly correspond to the colors red, green, and blue. When you see a color, each cone sends its own distinct signal to your brain. For example, suppose that yellow light, that is, real yellow light with a yellow frequency, is shining on your eye. You don't have a cone specifically for detecting yellow, but yellow is kinda close to green and also kinda close to red, so both the red and green cones get activated, and each sends a signal to your brain saying so. Of course, there's another way to activate the red cones and the green cones simultaneously, if both red light and green light are present at the same time. The point is, your brain receives the same signal regardless of whether you see light that has the yellow frequency or light that is a mixture of the green and red frequencies. That's why, for light, red plus green equals yellow. And how come you can't detect colors when it's dark? Well, the rod cells in your retina take over in low light conditions. You only have one kind of rod cell, and so there's only one type of signal that can get sent to your brain, light or no light. Having only one kind of light detector doesn't leave any room for seeing color. There are infinitely many different physical colors, but because we only have three kinds of cones, the brain can be tricked into thinking it's seeing any color by carefully adding together the right combination of just three colors: red, green, and blue. This property of human vision is really useful in the real world. For example, TV manufacturing. Instead of having to put infinitely many colors in your TV set to simulate the real world, TV manufacturers only have to put three: red, green, and blue. Which is lucky for them, really.
Okay, so the last thing we're going to do in this video is have a look at the last dot point, outline the role of rhodopsin in the rods. So the visual pigment rhodopsin present in rods consists of a protein molecule called opsin combined with a simple light-absorbing part called retinol. Retinol is a derivative of vitamin A. If vitamin A is lacking, vision is affected and a condition known as night blindness may result. So you need to make sure you eat your carrots. The main function of the photochemical pigment rhodopsin is to absorb light. When light strikes the rhodopsin pigment, the light energy is absorbed and rhodopsin changes from its resting state to an excited state. This change is due to the activation of the retinal part of the rhodopsin. When light strikes the rhodopsin, the retinal becomes activated. This causes rhodopsin to split into its protein opsin part and the free retinal part. The split rhodopsin pigment is said to be bleached, but the change is only temporary. A series of biochemical steps follow, whereby the activated pigment causes a change in electrochemical charges of the membrane of the cone of the photoreceptor. This is the start of an electric impulse that moves along the receptor, triggering the release of a chemical substance known as a neurotransmitter. The neurotransmitter then stimulates a bipolar cell, generating an impulse in this cell. The signal is termed electrochemical because it involves both an electrical change in the membrane and a chemical release of the neurotransmitter. The bipolar cell transmits the electrochemical signal to the ganglion cells, which in turn carry the signal to the brain. So that we've looked at this a little bit already in looking at how the light gets from the retina to the brain. So rhodopsin, the photochemical pigment which was temporarily bleached or broken down in the presence of light, is then regenerated so it can be reused. The retinal and opsin recombine with the help of enzymes. This allows a new image to be received, and the presence of vitamin A is essential for these steps to occur. So that's it for this video. We'll be doing some activities in class that look at these different concepts that this video covers.